Hi, hello everyone. Please be seated. It's time to start. It's my pleasure to introduce Dr. Jeiicho Fukuda. He's from Keio University. He's chairman and professor in the Department of Cardiology. He will present today is IPS cardiomyocyte generation, selection, and delivery. Thank you. It's your turn.
Thank you very much. I am Jeiicho Fukuda. I had just retired a professor of the Department of Cardiology, Keio University, just 2 months ago, and became a CEO of BioVenture Heartseed. Today, I'd like to talk about preclinical evaluation and clinical trial of human iPS cell-derived purified ventricular cardiomyocyte transplantation for the treatment of severe congestive heart failure. This is my disclaimer. You know, loss of cardiomyocyte often causes heart failure in various types of heart disease. And you know, the cardiomyocyte cannot regenerate by themselves. So in order to rescue the heart failure, we needed to supplement the cardiomyocyte. Thus, we manufacture the regenerated cardiomyocyte using human iPS cells and transplant them into the failing heart. The final goal of the regeneration therapy is like this. This slide shows multiple steps for regeneration of the heart. In order to achieve heart regeneration for the treatment of severe heart failure, we needed to overcome multiple steps. Efficient generation and safe and high-quality iPS cells, massive expansion of iPS cells, selective induction of ventricular-specific cardiomyocyte, purification of cardiomyocyte to eliminate residual iPS cells and non-myocyte, development of the efficient cell transplantation method, generation of GMP-level cardiomyocyte production systems, and preclinical animal experiment to confirm safety and efficacy. Actually, we had overcome all of these steps, so we began the clinical trial. As you know, there are several types of cardiomyocyte: pacemaker cells, atrial and ventricular cardiomyocyte. Myosin light chain 2A is an atrial cardiomyocyte marker, and myosin light chain 2B is a ventricular cardiomyocyte marker. Actually, we can separately induce both atrial and ventricular cardiomyocyte. In this experiment, we induce ventricular-specific cardiomyocyte. Some of the cardiomyocytes were also red-positive cells, but these red-positive cells also express MLC2B, indicating these are the fetal ventricular cardiomyocyte phenotype. Action potential recording revealed that the shape of the action potential is very similar to that of ventricular cardiomyocyte, and isoproterenol increases the beating rate. And ES and iPS cells are one of the most attractive cell sources in regenerative therapies for patients with heart failure. However, if non-purified ES or iPS cells-derived cells are transplanted, they will form a teratoma like this. So elimination of the residual stem cells and purification of cardiomyocytes are necessary. This slide shows the ideal strategy for cardiomyocyte purification. Red beating cells are cardiomyocyte. Sky blue indicated undifferentiated cells, and the other colors indicated non-myocyte. If we can make a medium in which undifferentiated iPS cells cannot survive, or non-cardiomyocyte cannot survive, we can purify the cardiomyocyte. In order to achieve this, we spent 8 years. As Dr. Shugo Toyama presented, we previously reported that depletion of glucose and glutamine and supplementation of lactate can purify cardiomyocyte. This is iPS cells. Glucose and glutamine are two major energy sources for iPS cells. These two compounds is definitely necessary to maintain iPS cells. If we deplete glucose and glutamine from the cell culture medium, iPS cells cannot survive. In contrast, cardiomyocytes express lactate transporter, and cardiomyocyte can uptake lactate, which was changed into the pyruvate by LDH. You know, LDH is expressed in the cardiomyocyte and are used in the mitochondria to generate ATP. So depletion of glucose and glutamine and supplementation of lactate can purify cardiomyocyte. Left-hand panel indicated before purification. As you can see, the beating cells are cardiomyocyte. The cells surrounded by beating cardiomyocytes are residual iPS cells and non-myocyte. If we deplete glucose and glutamine from the cell culture medium and supplemented lactate, only cardiomyocyte can survive. So using this system, we can produce a highly purified cardiomyocyte. Next step is a cell transplantation. We transplanted iPS cell-derived cardiomyocyte after enzyme digestion or thawing the frozen cardiomyocyte. But this floating single-cell transplantation system is not good. Very limited number of transplanted cardiomyocytes survived following the flowing single-cell injection. Most of the cardiomyocytes will die or lost by backflow due to bleeding. So we collaborated with some chemical company to make this specially designed dishes. And a right-hand panel indicated the cardiomyocyte spheroid. We investigated how to increase the cell survival after transplantation. And we finally found that cell aggregation can strongly increase the cell survival after transplantation. So we call this cardiomyocyte spheroid. Each spheroid contains 1,000 cardiomyocytes, and they strongly beat like this. The upper panel indicated the mouse ear cell-derived purified cardiomyocyte spheroid transplantation into the mice heart. This is 3 weeks and this is 8 weeks after transplantation. As you can see, this large striated muscle is adult cardiomyocyte. But you can find small cardiomyocyte. This is transplanted, newly regenerated cardiomyocyte. Cell aggregation strongly increases the cell survival after transplantation. Please take a look at the size of the cardiomyocyte. This is 8 weeks. As you can see on the right-hand panel, this is pre-transplantation, 3 weeks and 8 weeks after transplantation. Green indicated cardiomyocyte nucleus and red indicated cytoplasm by actinin. And the transplanted cardiomyocyte shows physiological hypertrophy after transplantation. The size becomes bigger and bigger after transplantation. Lower panels indicated human ear cell-derived cardiomyocyte spheroid transplantation into immunodeficient mice heart. In this case, human cardiomyocyte marked with red dye. And this is the result. As you can see, the human cardiomyocyte can survive in the immunodeficient mice heart and showed beautiful striation. In this experiment, we transfected respirase gene into the iPS cells and induced cardiomyocyte. The upper panel indicated single floating cardiomyocyte transplantation. As you can see, these fluorescence indicated chemical fluorescence indicated a number of surviving cardiomyocyte. Lower panel indicated cardiomyocyte spheroid transplantation. We transplanted the same number, but as you can see, there is a big difference. The right-hand panel indicated a quantitative analysis. So we can strongly increase the cell survival by transplanting the cardiomyocyte spheroid. This is the immunostaining of the human iPS cell transplanted cardiomyocyte. Graft area is a human cardiomyocyte, and host area is a mouse. Left-hand panel indicated 2 weeks, and right-hand panel indicated 4 months. The size of the cardiomyocyte is very small in 2 weeks. However, the size of the transplanted cardiomyocyte shows bigger and bigger after transplantation. This is higher magnification. So transplanted human iPS cells showed physiological hypertrophy and sarcomere development. Next slide indicated the immunostaining of the transplanted human cardiomyocyte into mice heart. In this case, red indicated ventricular cardiomyocyte, and green indicated fetal ventricular cardiomyocyte, atrial cardiomyocyte. At 2 weeks, small fraction of the transplanted cardiomyocyte was stained with green. However, at 6 months, all the transplanted cell shows in red. The right-hand panel indicated a quantitative analysis. These findings clearly indicated small fraction of transplanted human iPS cell-derived cardiomyocyte showed ventricular phenotype at 2 weeks, but fully differentiate into adult phenotype at 26 weeks. So we transplanted human cardiomyocyte into nog mice heart, and immunostaining was performed. In this case, brown area is transplanted human cardiomyocyte. As you can see, these are the higher magnification. The transplanted human cardiomyocyte showed physiological hypertrophy and an adult phenotype. Beautiful striation can be observed. No scar formation was found in the transplanted area. This is 4 months after transplantation. The right-hand panel indicated 1 year after transplantation. In this case, we transplant human cardiomyocyte into the nog mice skin. As you can see, the transplanted human cardiomyocyte showed spontaneous beating, and they formed a completely heart tissue. And very interestingly, neovascular formation was observed, and this newly developed vessel supplied the blood flow into the beating heart. Next, we developed an original cell transplantation device. This is a specially designed needle. This is blind end, no bevel, cone-shaped tip, and 6 side holes. And we made this kind of several needle attached cell transplantation system. Please take a look at this, the right-hand panel. As you can see, using this specially designed needle, there are no bleeding after transplantation. So no beveled tip of the needle resulted in minimum damage to the vasculature and reduced bleeding. This experiment is performed in collaboration with Professor Shiba in Shinshu University. This slide indicated the effect of human iPS cell derived cardiomyocyte spheroid transplantation on cardiac function in cynomolgus monkey model. In this case, we transplanted 20 million human iPS cell derived cardiomyocyte spheroid into the left ventricular wall. dark brown area is transplanted human cardiomyocyte. Human cardiomyocyte can survive in the monkey heart with using the immunosuppressant. This is 90 days after transplantation. The right-hand graph indicated changes in percentage fractional shortening after human iPS-derived cardiomyocyte transplantation. Human iPS cell-derived cardiomyocyte transplantation group increased cardiac contractility. Next slide is a report from other researchers. The left-hand panel is a report from Yuji Shiba's group, and right-hand panel indicated a report from Chuck Murray's group. They used either iPSL or ESL-derived cardiomyocyte transplanted into the monkeys. And the transplant, they reported that the transplantation of cardiomyocyte improved cardiac function but caused arrhythmia, especially at the first 1 month. So graft arrhythmia will become a problem at first 1 month from transplantation. So how about our case? This slide shows the effect of human iPS-derived cardiomyocyte transplantation on cardiac arrhythmia in cynomolgus monkey model. We transplanted the same number of cardiomyocyte and observed the ventricular tachycardia by Holter monitoring. We transplanted 5 monkeys, and 1 monkey among 5 showed a 9-minute sustained ventricular tachycardia. But this is only 9 minutes. And other monkeys did not show any significant arrhythmias. The possible reason why arrhythmia is reduced might be as follows. Number 1, we transplanted ventricular-specific cardiomyocyte, and cardiomyocyte purity is more than 99%. Relatively small number at 1 injected site. High survival rate of transplanted cell. That's why less inflammation at the transplantation site. And the transplant needle with less vascular damage. These factors may contribute to reduce arrhythmia. So this slide shows a clinical time course of heart failure and timing of cardiomyocyte spheroid transplantation. As you know, heart transplantation is performed at stage D. So when is the best timing for supplementation of regenerated human iPS cell-derived cardiomyocyte? In my opinion, late stage C is the best timing for supplementation of cardiomyocyte. Based on these preclinical results, we begin the clinical trial, LAPIS study. This is a multi-center open label 2-group dose escalation phase 1-2 study. Objective is to evaluate the safety and efficacy of HS001. This is the code name of cardiomyocyte spheroid in patients with heart failure. 10 patients with severe heart failure due to ischemic heart disease will be enrolled. This program is developed by Heartseed and our collaborator Novo Nordisk. This is the design of the LAPIS study, phase 1-2 clinical trial. After screening, patients undergo HS001 transplantation. Patients take immunosuppressants and have efficacy and safety assessment. The first 5 patients will be treated by low dose of HS001. The last 5 cases will be treated with a high dose of cardiomyocyte. Key inclusion criteria is LVEF less than 40% and NYH functional class is equal or greater than class 2, and simultaneously with CABG. The primary endpoint is safety and tolerability by adverse event in the 25th week post-transplantation. Secondary endpoint is LVEF, myocardial wall motion, and myocardial blood flow. This clinical trial is non-randomized, sequentially assignment, and open label. 3 patients had already received transplantation of HS001. Unfortunately, I cannot show the results right now, but I have a very good feeling from the initial clinical trial. This is the take-home message. In the near future, human iPS cell-derived cardiomyocyte transplantation will be applied to the patients with severe congestive heart failure. Thank you very much for your attention.
Thank you, Dr. Fukuda. Now we open the questions.
Thank you very much, and congratulations to this achievement. I think it's really great that you are going so far. I have 2 specific questions. You said low dose, high dose. What exactly, I mean, how many cells do you apply?
Yeah.
And the second question is, you say that you have a mature ventricular phenotype and show the MLC2V, which is, I think, fine. But we and others have shown or have found that the switch from slow skeletal troponin I to cardiac troponin I comes later and is maybe in this respect a more robust marker of maturation. So I was wondering whether you did that as well.
For the first question, low dose is 50 million cardiomyocyte. High dose is 150 million cardiomyocyte.
Okay.
And second question is, how about the maturation, right?
Yes. Whether you use the switch from slow skeletal to cardiac troponin I.
Actually, in the in vitro condition, before transplantation, I mean, we try to maturate the cardiomyocyte as much as possible by adding some reagent. But basically, at the timing of transplantation, not fully maturated. But this timing is very good because fetal or neonatal cardiomyocyte, they strongly use lactate as an energy source. So this is very good for transplantation. But I don't know the exact state.
So thank you very much, and also congratulations to the move to the clinic. The question I have is related to the patient population. So you are doing that on top of CABG surgery. Why do you do that on top? And would you be able to distinguish the effects that are introduced by CABG versus the effects that are because of the cell implant?
Yeah, this is a quite reasonable question. And we performed meta-analysis of the cardiac bypass surgery to the low EF patient. And right now, the number of treated patients is only 3, so it is difficult to say. But only CABG can improve a very small percentage of ejection fraction. But we transplanted into 45 different sides of the heart, so in various areas. And I cannot say the results because lawyer prohibited. So
then I assume that later on you are aiming to do that percutaneously. So at the moment it's a surgical intervention, and,
yes,
and you're shifting then to a cath-based approach probably.
Yeah. There is, we can compare the low dose and high dose and control CABG. This is not simultaneously performed, but we have this database, so we can compare the historical control.
Okay, thank you.
Okay, thank you. We move to our next speaker is Sanjay Sinha, and he is a professor in cardiovascular regenerative medicine at University of Cambridge. And welcome MRC Cambridge Stem Cell Institute. And thank you.
Great, thank you for inviting me. I'm delighted to be here in Kyoto for this fantastic meeting, and it's a real pleasure. So I'm going to talk about the epicardium, the role of this tissue lineage, and how it can be used to augment cardiac regeneration. So epicardium is this epithelial structure on the outer surface of the heart, and it's very important during development. We know that a number of cell types come from the epicardium, so smooth muscle cells and fibroblasts come from the epicardium, and epicardium also has extensive crosstalk with developing cardiomyocytes and blood vessels. Now, in the adult heart, the epicardium becomes quiescent, and the markers of epicardium, the WT1 and TCF21, are downregulated. But after cardiac injury, we see reactivation of the epicardium in the adults, and we see a limited epicardial-derived cell migration and limited angiogenesis and limited cardiomyocyte proliferation in adult mammals. And of course, this is part of the problem. It is, however, necessary for cardiac repair, particularly in organisms where we do see great cardiac regeneration, such as the zebrafish. This is work taken from Ken Poss's group, where he ablated the epicardium in the zebrafish, and you can see that in the normal zebrafish you get great cardiac regeneration after injury in the zebrafish, but if you ablate the epicardium, you're left with a scar. So epicardium is necessary for regeneration in this model. So in my group, we've generated epicardium in vitro from pluripotent stem cells. We published this work some time ago, and we get really nice looking sort of a cobblestone pattern of epicardium in vitro. And this can be pushed to generate fibroblasts and smooth muscle cells. And if we inject this into the circulation of a developing chick embryo, then the epicardial cells, the human epicardial cells, will home into the right location. And you can see here they'll go into the cell epicardial space and continue to express their epicardial markers. So it's a nice bona fide model of epicardium. I don't need to tell this audience the sort of problems with cardiac regeneration. We know that although many animals can regenerate their heart, mammals can't, and this is the scale of the problem. We're looking at about 1 billion cardiomyocytes that we need to replace. And this approach of generating cardiomyocytes from pluripotent stem cells and injecting them in has been quite successful. And we just saw some examples just now. This is an image from the Murray group. But of course, this has a number of problems. One is the cell survival and retention with this approach. The other is that many studies have been done in acute injury models, and actually what the patient group were looking for is the chronically infarcted heart with a very scarred substrate. And finally, there's a challenge of integrating the donor cells with the host myocardium, and I think this is a real issue. Our potential solution to this has been to combine the use of epicardial cells with cardiomyocytes so they can recapitulate their developmental role in terms of promoting cardiomyocyte maturation and proliferation. And we've shown previously that in a athymic rat model, we can get really nice cardiac grafting, and the grafts are much bigger when we incorporate epicardial cells than if we just use cardiomyocytes alone. In fact, they're 2.5 times bigger. And the delivery of epicardial cells leads to profound vascularization as well within the graft. And we've shown previously as well that that also helps to improve function, so that the combination of cardiomyocytes and epicardial cells work much better than cardiomyocytes alone, which works better than just the epicardial cells or no cell therapy. What we wanted to do then was to take this forward, first of all to ask what are the molecular determinants of epicardial function, why is the epicardium having this beneficial effect, and also how does this combined cell therapy that we're using work in the chronically infarcted model. So we collaborated with the Murray group. We used the rat infarct model. This time it has an infarct. It was 60 minutes of ischemia reperfusion, and then we wait a month. And the rat, this gives you a nice scarred, damaged heart at that point. We then inject with cell therapy, with a combination cell therapy. We have a no-cell negative control. For the positive control, we wanted to use a cell type that we thought would give us good regeneration, and we wanted to overcome any potential species issues. So we used rat neonatal cardiomyocytes that are isogenic with the rats that are receiving the grafts. And what we saw is when we put the neonatal rat ventricular cardiomyocytes into the rat model, we got really nice large grafts, and these matured and looked really, they were very mature. I mean, you couldn't tell the remote region from the grafted region. The only way we could tell this morphologically was because we'd used male-female mismatching, so we could stain for ZIS, which is these red stains. So you can see these are female cardiomyocytes into male recipients. And we can see really nice cell-cell junctions, these gap junctions here staining for connexin 43, really nice junctions between the donor cells and the recipient cells. So we're really struck by how well, if you put isogenic matching cells in, these would graft into even in a chronically infarcted model. When we put the human cells in, we saw reasonably large grafts as well. They didn't look as mature 3 months after injection as the rat cells did. You can see that there's connexin 43 expression, but they don't make, there was some evidence of nice gap junctions, but not as well formed as in the rat cells. And in terms of integration with the host, there was quite a difference from when we put the rats. There are some cells where we do see, in the red you see the human cells, in the green are the host rat cells, and there are some cells where we see nice gap junctions between the host and the donor cell. But for the majority of the cases, we didn't see good integration. So what about function? Well, without any cells, after you see continued deterioration of this rat function after 1, 2, or 3 months. Treatment with either rat cardiomyocytes or the human epicardial cardiomyocyte combination prevented that deterioration, and there was a significant difference between the cell therapy groups and no therapy. We couldn't statistically tell any difference between these groups, although you can see there's probably more variation when we injected the human cells in compared to when we put the rat cells in. We also saw revascularization, neoangiogenesis within the graft. When we stained for CD31, both within the rat ventricular cardiomyocyte grafts and the human cardiomyocyte grafts, we saw a reasonable amount of neoangiogenesis. And in fact, there was greater neoangiogenesis. This is just preliminary data from a small number of animals. This work is still ongoing. But we see greater angiogenesis both in the graft and in the remote region when we put the combination of epicardial cells and cardiomyocytes in compared to the neonatal rat group. Okay, so how is epicardium having this beneficial effect? We've seen before, both in engineered heart tissues as well as in the infarct model, that we see a lot of fibronectin deposition when we put epicardial cells into these models. We then looked a little closer at fibronectin. We did some loss of function studies. This is just in engineered heart tissues. First of all, we knocked out fibronectin in epicardial cells and then made the engineered heart tissue. And you can see in red cardiomyocytes alone, when we add epicardial cells, we see a significant inotropic increase in the function of the engineered heart tissues. But if the epicardial cells have fibronectin knocked out, we lose this benefit, and it goes back down to the level of just cardiomyocytes alone. We did this again, this time using an inducible short hairpin RNA, that knocks down fibronectin rather than knocking it out. And again, we see the same effect. We see a benefit of contractile benefit when we add epicardial cells to an engineered heart tissue, which is ablated when we knock down fibronectin. Then we went and asked the reverse question: can we supplement fibronectin and replace the use of epicardial cells, which we did with 3 different concentrations of fibronectin, 10 micrograms per mil, 50, and 100. And actually, initially, we didn't get much of a response except for the highest concentration of fibronectin, which was then able to replace the function of epicardial cells. So fibronectin is necessary for the function of the epicardium. Whether it's sufficient on its own, I think, is doubtful. I think there's probably other things required. So we then looked, so this is just again looking at fibronectin, not the functional effects now, but on the structural effects on the myocardium. And we're looking at one of the parameters of structural maturation in engineered heart tissue. Here we're looking at sarcomere length. We can see the sarcomere length increases when we add the epicardial cells compared to cardiomyocytes alone. And this could be replaced if we use fibronectin instead of epicardium, even at a low concentration of 10 micrograms per mil. So for the structural effects, it seems a fairly low dose of fibronectin can substitute for the epicardium, but for the functional effects, such as contractile function, we need to add an awful lot of fibronectin, so there are probably other factors involved in terms of what the epicardium is providing. We then looked at, in more detail at the matrix that the epicardium lays down, and we decellularized the cells. This is looking at different types of matrix now, either from cardiomyocytes alone, the combination of the cells, epicardial cells alone, or HS27A cell, which is a stromal cell line. And you can see that with the epicardial cells, you get this very fibrous looking matrix, and this is under electron microscopy much more complex than what we see from the HS27A line. And the decellularized matrix from the epicardium, when we then used that and then put cardiomyocytes back onto it, we saw that it has the beneficial effect on sarcomere length compared to, for instance, either vitronectin, matrix from cardiomyocytes, matrix from HS27A cells. If we start looking at functional capacity of these cells, this is again just looking in 2 dimensions now on cardiomyocytes plated on this decellularized matrix. We looked at their calcium dynamics. We looked at time to peak. The time to peak on the HS27A cells was quite high, and then it was much lower when we used the epicardial matrix. This seemed to be lower than the other groups, but again, this was not statistically significant. So maybe not quite as much benefit functionally as we see structurally. We looked at cell adhesion. I think that's probably important if we're going to get cell attachment and survival when we inject these into the damaged models. And we looked initially at early time points. There wasn't much difference in the groups. But at 24 hours, we can see that cells placed on Matrigel and on the decellularized epicardial matrix were able to attach better than the other groups. And certainly by 48 hours, the epicardial matrix was better than Matrigel in terms of cell adhesion. Epicardial decellularized matrix also increased cardiomyocyte proliferation. This is just looking at EDU staining, and this is alpha-actinin on top of that to make sure we're looking at cardiomyocyte proliferation. And what we see is that the epicardial matrix promotes cardiomyocyte proliferation compared to other groups such as Matrigel collagen or the HS27A matrix. We've now done proteomics on this, and we're analyzing that to try and get to the bottom of that, but that's still work that's in progress, so I don't have the results of the proteomics to share with you at the moment. But we're going to try and tease apart exactly what those components are. So just to summarize, what I've shown you is that the stem cell-derived epicardium promotes cardiomyocyte function in vitro, potentially through fibronectin, and likely through other components as well. It promotes cardiac regeneration in vivo, and certainly in a chronically infarcted heart, which is always quite a challenging environment. But I think what I want to make sure flag really is that cardiomyocyte integration, or donor cell integration into the host heart, remains an issue that I think needs to be addressed. I want to acknowledge the people who have done the work and those who have funded this. Thank you very much.
Thank you, Sanjay. Beautiful talk. Now we open the questions.
Yeah, super interesting, Sanjay. Just wondering, I mean, fibronectin is produced by a lot of different cells. So for me, it's a little puzzling that fibronectin becomes kind of a limiting factor. So it's probably not only epicardial cells that produce fibronectin, right?
Yeah, I mean, you're absolutely right. After injury, looking now at the chronically infarcted model, we see we see fibronectin produced quite in excess, I think, by the epicardial cells. So I think it may be a quantitative difference. Certainly fibronectin is necessary, but I don't think it's sufficient. I think there are other things going on as well to really give us the full benefit of the epicardium. I mean, we see good benefit in terms of structure, but not as much benefit in terms of function just from the fibronectin. So I think there are other things going on which we need to get to the bottom of.
And the second short question, actually, what happens to the scar tissue in your chronically injured hearts? Is the scar tissue pushed aside or still present in between the transplant?
The scar tissue is still present. We inject both within the middle of the scar tissue and at the edges of the scar tissue when we do the injections, so there are 3 separate injections. The scar tissue is still present. We don't see resolution of the scar, and I think, so it's still there.
Okay.
Oh, thank you. Great talk. I'm very impressed. I have one question. What do you think is the superiority of epicardial cells over cardiac fibroblasts when transplanting with cardiomyocytes?
I'm sorry.
What is the difference of cardiac fibroblasts and epicardial cells?
Yeah, cardiac fibroblasts obviously originate from epicardial cells. We haven't compared cardiac fibroblasts. We've compared mesenchymal stem cells, we've compared the HS27A line, and the epicardial cells are as good or better than any of the other cell types that we've compared. The reason we haven't compared cardiac fibroblasts is because I don't think it's a very feasible cell type for cell therapy. So stem cell-derived epicardial cells would be very feasible for a cell therapy source, but I think cardiac fibroblasts, certainly primary fibroblasts, wouldn't be, so we haven't done that direct comparison.
Okay, I understand. Thank you very much.
Great talk. I was curious to know whether or not you thought about the workload hypothesis. Often the epicardium has a very high workload. We've previously published that the phosphorylation status of myosin light chain 2V is often higher in the epicardium, and when we annihilate the gradient that occurs, you often kind of get deterioration of workload and torsion. And I was wondering whether you looked at that as a potential mechanism for why, epicardial-based cells may have this benefit.
No, this is really interesting. It's not something we'd looked at, so I might come and talk to you more afterwards.
Okay, thank you.
Yeah. With or without the fat? Yeah, so these are stem cell-derived epicardium, so we don't get any fatty tissues.
Okay, and so what do you think the mechanism of the increased angiogenesis is?
Yeah, that's a really great question as well. So I think this might partly be the fibronectin, but we've also looked at paracrine factors, both exosome-related and non-exosome-related paracrine factors, and we have found that extracellular vesicles certainly promote angiogenesis. So the epicardium releases extracellular vesicles, which are pro-angiogenic. We've been investigating the sort of microRNA components of that, and we've identified some of the microRNAs that contains. So that might be one factor that promotes the angiogenesis. The other factor might be fibronectin. There's actually quite a lot of fibronectin attached onto the surface of the extracellular vesicles. So how the fibronectin is delivered is an interesting thing to think about. But I think it's a combination of different mechanisms that leads to increased angiogenesis.
Thank you very much.
Thank you. And now we move to our next speaker, Tom Eisenhagen. I hope I pronounced it right. And Tom is a director and professor in the Institute of Experimental Pharmacology and Toxicology, University Medical Center, Hamburg, Eppendorf.
Thank you very much. I'd also like to welcome, to thank Jay and Keiichi for the invitation. It's really great to be here. You got everybody here. In addition, it's always nice to be in Japan. So I come back to a subject which was touched upon by Wolfram, and so I think I can go through some things pretty quickly. Just to say, we have a strategic partnership with Evotec to develop this cardiac repair approach, and I have some affiliation with Dynacore. So after this phase of autologous stem cells with more or less good results, I would say, we are now at a situation where I think there is realistic hope that you can regenerate the heart by either giving reprogramming factors or pro-proliferative factors, or, and that's something I will discuss about, giving cells directly, either as patches or as cell injections. And somehow we went a step back and tried to answer some basic questions, and they're shown here. Do we really need new heart muscle to repair, to improve the function? If yes, how much? Do grafts really actively contribute to contractile function? And what are the optimal means to regenerate large coupling grafts? And since we have a history with EHTs, which goes back almost 30 years now, we had, of course, a bias towards the engineered heart tissue. I think if you see pictures like that, and this is just a summary of what has been published by us and others, here there's some results with patches, and here are results with pigs and 3 non-human primate, and I think you can easily always recognize the graft. I think it's fair to say that in the meantime, the size of the grafts is at a scale where you can say, well, that has a realistic chance really to improve function. Question is, how much do you need? And Jeiicho Fukuda just gave us an example that maybe we need less, but if you use classical suspended cell injection, and this is mainly work of the Murray group and Laflamme, you see here 1 billion for 40-gram heart, 400 for 30, 750 for 40-gram heart, and that always results in about a 10% absolute increase in left ventricular ejection fraction. So if you summarize these cell injection studies, simple cell injection, you come up with, you need about 13 to 25 million per gram myocardium to get 10 or 15% replacement of the scar, and that is associated in general by improvement by 10% absolute. Okay, so let's see what the patches are doing. We are using this kind of patches. You see the size at about 2.5 centimeters by 1.5. And we are mainly working with a guinea pig because it's a pretty robust model. And so some years back, we asked the question, how much do we need? And the hope was that actually we need less than what we call the high dose. So we reduced both the concentration of the cells in the patches and the size of the piece we transplanted onto the guinea pig heart, coming up with 12 million, about 8 and 4. And the result was clear, even though a little bit disappointing for us. We received, or we induced the same scar size in all of these 3 groups, cell-free, dose 1, dose 2, dose 3. As expected, the graft size increased with the dose. You see a large scatter. And this is echo data, just one parameter, fraction area change. You see always before injury, after injury, and then 4 weeks later. No change, of course, here in the cell-free, no cell low, medium, and only the high dose, we saw what we were used to, an increase by, again, 10%. So the conclusion here is, well, we need also a lot of cells, maybe less, 5 million instead of 15 per gram, and that results in the same increase. If you calculate this number up to the human heart, you would need 1.5 billion. Wolfram told us a high dose in his studies is 800 million. We're in the same range here, but it's still an enormous amount of cells. We thought a little bit about applicability, so we produce now these, what we call giga patches. They are 5 by 7 centimeters, and you see they beat well and contain 500 million cells. I would say that's possible to think about transplanting these these patches. However, I think we were really interested in the question because this has never been answered. I mean, we are talking about cardiac repair, regeneration, but nobody ever showed that these cells really do what we expect them to do, namely contribute to an improvement in contractile function directly. You could say, well, that's trivial. I do not think it's trivial because there are so many studies showing benefit without any new cell. For example, the coupling, as we have discussed already, is an issue. And if it's not about contractile force, why do we need all these expensive and potentially dangerous cells? So Florian Weinberger in our lab perceived this study where we have this cryo injury. So we have here left ventricular function. This was the idea. The injury reduces a decrease in contractile function. Then 1 week later, we transplant the patches or we inject the cells. We get an improvement in function. And if this improvement has anything to do with the contractile force of the cells implanted, then a selective switching of contractility in this transplanted cells should lead to this red line. If it would be all paracrine, it would be green. And we have nice tools nowadays with these optogenetic tools. There are numerous. We use this one, which is IMO4. This is a chloride channel. If you open it, it depolarizes the cell. And it has two features. It has a luciferase on. So if you give a luciferase substrate, you produce local light, and this is opening the channel, or you directly shine blue light on these channels, and they open, depolarize the cell, and stop them immediately. That was the idea. So we integrated them in iPSC, made 2D cardiomyocytes here on the left, EHTs below. You see the EHTs, nice structure, alpha-actinine. And here in green, that's the channel, and you see it is nicely integrated in the plasma membrane, not only, but mainly, and you see the overlap here. So that looked good. And then we made EHTs and shone light on. So this is a spontaneous activity of EHTs in vitro. And then if you shine on these ILMO modified cells, or EHTs from them, blue light, you see it's immediate stop. I mean, it needs 2 contractions, 3 contractions, then it stops to beat. Then you cannot electrically pace them anymore because they are in a depolarization block. And then if you stop the blue light, they start to beat again after 5 seconds, more or less. And nothing happens, of course, in the wild type, which can be nicely paced. So we thought that is a good tool. So in this time, we injected the cells, not as a patch, but injected them, and you see, let's say, a representative result. You see here clearly the cells, the human cells in a guinea pig heart. Sometimes they are not exactly where we wanted them to be. That is how it normally looks. If you zoom into the tissue, you see nicely the injected cells in green because they are GFP positive and the host is not. And you see here the extensive zones of coupling. We always have this coupling issue. And I think we didn't prove coupling here, but at least there are extensive zones where there could be coupling. And if you zoom further in, you see connexin 43 positive areas. So we were quite impressed by this closeness. We never actually saw this with the patches, I have to say. And now this is the main result. You see, we took their hearts out, we subjected them to a Langendorf perfusion, measured with a catheter, left ventricle developed pressure. It was very stable, and then shining blue light on, you exerted this small but significant decrease in contractile force, and switching it off brought it back to normal, and nothing happened with red light, and nothing happened with wild type cells, etc. So that's it. That's a proof that contractile cells indeed participate. We saw quite some scatter, I have to say. And this is all the hearts here. There's a cohort of about 15 or so. And then if we separated the group in those which reacted to the light and those which did not, at least it was clear that the larger grafts were associated with a better response. So the conclusion here is, yes, we believe this is a proof that the force of the grafted myocardium directly contributes to systolic force generation in guinea pig. That is, the effect size depends on the graft size. Overall, the effect was small, but I would say realistic. And indirect effects, paracrine effects, probably contribute to the overall improvement. A secondary result of this study was we were surprised that in contrast to our expectations, because I said we had this patch bias for the EHTs, we were surprised that we saw pretty good grafts after cell injection. Look at the 12% scar, 20 million, this is 7.5 million per gram. This is somewhat in between the published numbers of Chakmari, Laflamme, etc., and our own patch data. And sometimes we see grafts like that. Look at that. This is amazing. Same dose here. This is a cross-section of a heart. You see this is host here, but all of this here, all of this is injected. All of this is human graft in this guinea pig. So sometimes you see huge grafts, much more than we expected. And they do mature quite well. You see here after 8 weeks, a very nice structure. And importantly, I told you about this switch from slow skeletal to cardiac troponin. So these are all cardiac troponin positive. So they do mature quite well. The statistics are that after 4 weeks, we have only 10% cardiac troponin I, and then it goes up to almost 30% and probably goes on later. So when we saw these results, we were, as I said, working with Evotec. We finally said there's more arguments, at least in our view, to go on with a cell injection. The EHTs probably really have no risk whatsoever in terms of arrhythmias, and I think probably because they don't couple, but also maybe a lower benefit, because if they don't couple really, it's difficult to explain the results. So that we are going now for single cell. Actually, after 30 years of EHTs, I have to say it's a bit painful for me myself, but it's like that. Arrhythmias are an issue. We talked about coupling. I think this is one of the few evidence that patches really can couple. This is in a few years ago in our studies. But these were only 2 out of 7 hearts. The rest did not couple. And on the good side, of course, if it doesn't couple, it doesn't induce arrhythmias. You know this. I think it has been shown already. In the cell injection studies in pigs, in the Laflamme or Murray group, you see here untreated pigs had a mortality of more than 60%. It could be reduced by amiodarone and ivabradine to 25%, but still, I mean, it is a problem. And I guess you are aware of the attempts of the Murray group to reduce this. So they knocked out 4 ion channels and overexpressed the 5th one. And then they got really almost no arrhythmias. But even here, there is some arrhythmias. So in this respect, it's very interesting that you did not see, Keiichi did not see arrhythmias, because I do think this is a problem. So the summary of my talk is here. Surprisingly, we found that the injection of these myocytes forms similarly large grafts in patch implantation. Injected cells couple electrically while patches do not. Cell injection bears arrhythmogenic risk that require novel therapeutic strategies, in my view. And I personally believe that clinical therapy on the long term, and I think Wolfram alluded to this already, really requires attempts to reduce the immunogenicity, and that's the concept of with our collaboration with Evotec. So we are going for cloaking, drug-induced kill switch, arrhythmia prevention. All of this will probably require, I mean, this is heavily worked on already. This is something which we are working on now. And on the right side, they invested a lot of energy and actually successful to set up large-scale, completely 3D bioreactor-based differentiation, and this looks extremely good. Here it's turned out that single nuclear sequencing is extremely powerful to see contaminations, and it's going to be less than 0.0 something %. And with this, I close and thank the people, is particularly Florian Weinberger, who spearheaded these activities, and Tim Stüdemann, many other people involved, and also people, also fundings. And I thank you for your attention.
Thank you. Now we open the questions.
Thomas, great talk. I'm really struck by how well your cells integrate with the guinea pig heart, more so than we see in the rat. Do you think this is because of a difference in heart rate, or do you think it's an immune issue?
This is always the why questions. First of all, I don't know, but indeed, as you say, we chose the guinea pig for mainly the reasons of a relatively human-like cardiac physiology. It had a wide action potential, it had a heart rate around 250. So we always thought that this is a better conditions for coupling, but it remains speculative. I was surprised about this intense coupling. I mean, this is not really, it's structural integration, but it's extremely good.
Coupling, yeah.
Yeah.
Hi, Thomas. Sorry.
Oh, go ahead.
Thank you. Excellent data, and I have a few questions from a basic scientist perspective. So maybe you shared it and I missed it. So did you make those iPS cells into single cell suspension before they were injected into the heart? And the second question is, from human heart perspective, it would be a lot of cells, like billions of cells. So do you have any estimation of how many cells are integrating and how many cells are discarded from the heart into like bloodstream and things like that?
Yeah. Well, some of this I think I answered. So for your first question, we do differentiate cardiac myocytes from iPSC, then collagenase, isolate them by collagenase treatment and then inject them directly. And more or less what everybody else is doing as well. Why we do have quite this high efficiency, I cannot tell you. I mean, we are not the most experienced cell injectors, and I would have expected very bad results. I was super excited to these good results. We are using this pro-survival cocktail, but this is something which Chuck Murray developed, Michael Laflamme, not us. So otherwise, nothing special. And with regard to your first question, as I said, with the patch which had at that time the highest efficiency, I think, in terms of input-output, we needed 5 million cells per gram heart. And that ends up in 1.5 billion if you just linearly bring it up. Now we heard from Jeiicho Fukuda that they use 50 to 150 million. And in the end, it's all the question, how much graft do you get per input? And the data are the data at this point. I think there are several strategies to improve it. One is the spheroids. Others are, for example, pro-proliferative interventions. There are many other things. So I think that's not the end.
Thank you.
So I think the question of coupling is of course really important, but also pretty open. I mean, of course, cardiomyocytes can couple if injected. I mean, this has been demonstrated quite some time ago. Whether they contribute to contractile performance, I think this is nice data and very convincing with the on switch. But if you think about an injection into a scarred ventricle or patching onto the heart, doesn't really matter. You would in any case not have perfect coupling. So at least for some time. And I think this is one of the challenges that we all have, and it's just more a comment than a question. I have a question also, and the question is more related to the cardiomyocyte phenotype. They seemed relatively small still. So they seemed elongated and
thin.
And thin. So have you seen them develop over time into bigger cardiomyocytes, or is this just because of the time duration being too short?
I would agree, I mean, from this picture at least, that they look thinner than normal. What we saw, we checked 4 weeks and 8 weeks. And over this time, the maturation, as I showed with the cardiac troponin I, clearly increases, the length increases, and the width, actually, I'm not sure whether they evaluated that yet. Yeah. And whether this is really representative. What is clear is the orientation of the cells get much better over time. And with regard to your first thing, I mean, we are in the same boat, but I gave about 100 talks about EHTs, and they always ask me, but how do you want to explain improvement in function without coupling? Because nobody except us, I think, really showed coupling. We showed a bit of coupling, others showed no coupling, and I always had a hard time. And I just think that cell injection probably has a higher chance of inducing coupling because you bring the cells better into the myocardium. That's
it could be, but it's still, of course, you know, since it's not organized, it will be chaotic coupling at some point in time.
Yeah, yeah.
And that has to be
absolutely. That's why I think
somehow. And this we don't know how this really happens. And if you, I think we have to think about some alternative ways of synchronization of implants because I just simply cannot envision that any implant, be it an injected cell or a patch, would couple perfectly and look exactly like ventricular myocardium in the normal heart. So,
but the time course experiments of the arrhythmias consistently show an initial peak and then a ceasing of these arrhythmias. So that makes me believe that there is indeed a quantitative. Yeah. It's a bit of belief because it has not been really shown, but everybody showed this decrease of arrhythmias over time.
Good, I mean, but KT also showed, and I think we all agree that 90% of the cells are dying in that time frame. So that may also explain why arrhythmias go away.
Hey, Thomas, we are having fun. This is a really important question because arrhythmia is lethal. It can be a serious consequence. So I'm so glad to see some real data coming out with the technology. So the blue light is just like any light technology. There's a penetration issue. But however, your graft have it's like finger cross, have the surface, have the inner layers.
Yeah.
So it's difficult to activate in the same situation.
Absolutely.
That's one complication. Two, you have arrhythmia, but you have re-entry induced arrhythmia. You have ectopic center arrhythmia. Those are two different mechanisms, and I'm really interested in to hear what is your strategy to engineer cell to decrease the
arrhythmia
risk.
Yes. Okay. First question. You're right. Absolutely right. I mean, this experiment was not heroic like a clinical study, but it was also not easy because initially we didn't see anything. The blue light penetration in the tissue is indeed very, very slow. So what we are doing is we are underestimating the effect. We are underestimating it because if we would have reached it all, it would probably be better. The red light, which goes into the tissue much better, much deeper, clear, has no effect. So that's to your first sequence. So I fully agree, but this is underestimating the contribution to overall contraction. And the second question, how do we want to get rid of that? I mean, the point is we never saw any arrhythmias because we are always working with guinea pigs. So that's as a confounder. But we have good ideas, I would say.
Yeah, we can have one more short question, short answer.
Well, thank you. It's less a question than a brief technical comment. It turns out that clinically I do have experience with both techniques, the cell injections and the patch. And the point I wanted to make is that if you're considering clinical applications, and in particular multi-center trials with different investigators, the advantage of the patch is that it really allows to have a much more homogeneous and reproducible delivery. It's tough. Even with transepicardial injections, as we did, or endocardial injections that Joe is doing, it's more challenging to have an homogeneous, reproducible delivery of the cells from one injection to the other and from one center to the other. And I think that this heterogeneity might be a confounding factor in the interpretation of clinical outcomes.
Since you did these studies and me not, I don't comment.
Thank you. Thank you for all the discussion and the comments. Now I move to the moderator, to Dr. Fukuda.
So let's move on to the next presentation. The next presenter is Mauro Giacca, professor at the School of Cardiovascular Medicine, King's College, London. The title of his talk is Induced Cardiac Regeneration by Cell Cycle Activation. So Professor Mauro, please.
Thank you very much, Keiki. Thank you very much, Jay. It's really an honor and privilege for being, to be here with you today. These are my disclaimers. I'm working with 3 startups to do some of the work I will going to briefly show you today. We have to acknowledge one fact that as a community we have failed to run the revolution that in all other fields of medicine the introduction of biological therapies have made. So for the heart we don't even have a recombinant protein. Recombinant protein started with insulin in 1982. We have nothing for the heart. We don't have a monoclonal antibody. Even less, we have a nucleic acid, a vector, a cell, or a tissue. So it is surprising that one of the diseases that is the most prevalent in the world don't have any biological therapy yet. The kind of biological therapies we are attracted very much are small non-coding RNAs, and there are 3 categories of these. The oldest one to be introduced are antisense oligonucleotides. These have been around for more than 30 years, but have now really gained momentum because there are chemical modifications that can be introduced in these molecules that make them effective and stable once injected. The second category are RNA interference therapeutics, so siRNAs and and microRNAs, which work inside the cells by coupling with the RISC complex for activation of the strand that then inhibits messenger RNA. And the third categories are aptamers, and so conformational RNAs that bind to a target through their conformation rather through sequence. This is not the future, it is a present of medicine. There are already 13 of these therapies approved for clinical use, 9 antisense oligonucleotides and 4 siRNAs, which have been approved by the FDA, the EMA, or both for therapy. The kind of small RNAs we are very much interested in are microRNAs. I love these molecules for the same reason why sometimes you hear, well, microRNA will never be therapeutic because they target too many genes, so they have off-target effect. MicroRNAs don't have off-target effect because they target physiologically many genes, and we have to turn this into our advantage by targeting tens or hundreds of genes. These are molecules that can really impart the cells a desired phenotype. Think, for example, cardiomyocyte proliferation. You have this huge cell in which you need to dismantle the sarcomere, activate the transcription of the cell proliferation genes, change the kind of metabolism, and so on and so forth. It's very difficult to think that a single molecule with a single specific effect can do that. MicroRNAs are fantastic because they do complex functions. There are many, but not too many, approximately a bit more than 2,000 microRNAs, and so this means that you can obtain them as synthetic molecules and you can screen them for function in the laboratory, and they are small, so can be obtained as synthetic molecules and delivered. The beauty of screening is that you can search for a specific function, and you can search it independently what is the function of a microRNA in the tissue. Most of the microRNA we show you today are not microRNA that work in the heart physiologically, are molecules that we use pharmacologically to impart a desired function. This is a high-throughput screening facility that we have at King's College with libraries corresponding to RNA interference molecules for the whole genome of mouse and humans. And we started this adventure a few years ago by asking ourselves whether there are microRNAs that stimulate endogenous cardiomyocyte proliferation. So we screened the library of human microRNAs, ended up in finding a few that are particularly active at driving proliferation of mouse, human, rat cardiomyocytes. We come from the gene therapy field, so we have a lot of experience in AV vectors. So for us, it was quite obvious to develop an AV vector that contains the pre-mRNA gene for two of these, 590, 199A, and observe that if you take an AV vector expressing either of these two and inject it in the heart, you transform very big infarcts, as you see here, into much smaller infarcts. And by looking at the molecular reasons, there are all the correlates of cardiomyocyte proliferation and cardiac regeneration. We have now extended these screenings. We screened genes coding for the cytoskeleton, sarcomere 400, the siRNAs. We have a siRNAs that stimulate markedly cardiomyocyte proliferation in neonatal cardiomyocyte. We screen the ubiquitin proteasome system. We have one particular E3 ligase that is massive at stimulating cardiomyocyte proliferation. We screened all metabolic enzymes. Also here we have a few siRNAs against metabolic enzymes that stimulate proliferation. And so this is just to say that really it seems that manipulating the RNA interference pathway, you can achieve proliferation. 199A was also tested in large animals. And basically this is again an AV vector injected in 20 locations around the border zone, about 3 milliliters of vector. And the results seen by MRI after 1 month were really beyond our expectations. So a strong reduction of infarct size, a strong increase in ejection fraction, and remuscularization, so big scars in the control animals, much smaller scars, and presence of more cardiac muscle, which can be seen also here. You see this is an animal after 1 month in a control, dilation of the left ventricle, thinning of the ventricle, of the septum, and you see here there is much more muscle and the heart doesn't dilate. Unfortunately, however, you cannot use AV vectors to deliver a proliferative microRNA because these vectors express their transgenes forever. There is no way in a clinical setting of controlling still transcription of a transgene from a vector in a pharmacologically inducible manner. Bottom line, this microRNA keeps being expressed, and if there is regeneration after 1 month, from 2 months onwards, you start seeing the appearance of a cluster of highly proliferating cells, Ki67 positive, cells that express antigens that should not be there, like GATA4, which express very high levels of during embryonic development, or antigens that are never expressed in the heart, like myogenin, which marks rhabdomyosarcoma in skeletal muscle. And besides proliferation, this could be due also to the fact that with an AV vector, you cannot control the strand of a microRNA because you have to deliver the pre-microRNA gene. And so it is cells with its risk complex that decides which of the two strands are expressed. In the case of 199A, both strands are expressed equally, but the 3P is the pro-regenerative, the 5P has been reported to have detrimental effect. So we have to deliver these small RNAs as synthetic molecules. And over the years, we've tried very many different non-viral systems for delivery. So we try different types of polymers. We try cyclodextrin, 3 types of dendrimers, gold nanoparticles, carbon nanotubes. Nothing worked. I mean, coming from viral gene therapy, my conclusion were very reinforced that nothing is as good as viruses to do gene transfer. Until we stumbled upon the SNALP technology, so the stable nucleic acid lipid particle technology that permits the creation of nanoparticles, of lipid nanoparticles, that have approximately 80, 100 nanometers, and are formed of 4 lipids, 2 of which are helper lipids, usually cholesterol and phosphatidylcholine lipid. One is a PEG lipid, which gives stability to the particle and permits shedding from cells of the reticuloendothelial system when injected in vivo. And the 4th lipid is the most important one. It is a ionizable lipid, so a lipid that at acidic pH is positively charged and then becomes neutral when the pH is raised at physiological level. It is important to inject a neutral particle to avoid inflammation and to avoid activation of platelets and the coagulation system. And these particles become very popular now because the first has been approved for clinical use in 2018. It is patisiran for the delivery of an siRNA against transthyretin for liver amyloidosis, and then obviously for the Moderna and the Pfizer vaccines against COVID. We have worked a lot at improving these particles and finding particles that work well in cardiomyocytes by testing a number of ionizable lipids at different lipid ratios. We have now particles that we can inject in cardiomyocytes and drive proliferation, basically almost 80% of cardiomyocytes in culture conditions. We also worked at improving transcription of messenger RNAs, which will equally be delivered with these particles, with T7 in vitro transcription system, in which we have specific 3' and 5' UTRs that translate well messenger RNA in cardiomyocytes. And at the end, we have particles in which we can really deliver at more than 95% efficiency in cardiomyocytes. These are neonatal cardiomyocytes expressing the messenger and optimized messenger RNA for GFP. You can see basically that these are fantastic transfection reagents in cardiomyocytes, and they are fantastic transfection reagents also in vivo. This is a particle that we obtained in collaboration with Christian Planck, and it contains the messenger RNA for GFP. This time it is delivered directly in the left ventricle, 10 micrograms messenger RNA into 15 microliters injections. GFP here is developed with an antibody in red. You can see that you have the whole left ventricle free wall in mice that is completely transfected. And the same particle injected into the intramyocardial in pigs, this is a collaboration with Fabio Recchia in Italy, gives really transfection to large number of cardiomyocytes also in pigs. We have also worked by chemically optimizing the payload, so the microRNAs. MicroRNAs are difficult to be chemically optimized because you can work a lot on the passenger strand, but not much on the strand that risks you want to use. But we could introduce a series of modified nucleotides to increase stability of the microRNA for 12, 15 days in vivo, so sufficiently long to drive proliferation, but not too long to drive an unwanted effect. This is how one of these, our nanoparticles look like, so approximately 100 nanometers. You inject it in vivo, you stay there for 15 days, and a single injection is sufficient to drive regeneration in mice, so convert very big infarcts, like in these 4 animals, into much smaller infarcts by cardiomyocyte proliferation. We are now working with Jeiicho Therapeutics, a new startup I founded in London, to bring this to the clinic. While we were working on this idea of injecting small RNAs into the heart for cardiomyocyte proliferation, we came to the conclusion that this can be a very interesting strategy to not only to achieve cardiac regeneration, which would itself be a huge goal, but also to go beyond that, so to think at microRNAs as molecules that can transiently change the cell state and have for a few days make the cell to do a function that they normally don't do. then microRNA go away. You can titrate the time by which they go away by changing the chemical modification, but the cell regains its original properties. But in the meantime, you have achieved something. In the case of proliferation, you have achieved proliferation. But for example, there are other functions which you want a transient activity. For example, induce permissivity to AV vectors or stimulate gene editing. For AV vector, have been very successful in gene therapy. There are 18 products now approved by FDA, EMA in gene therapy. 6 of these are based on AV vectors. However, you have to inject 1 per 10 to the 14 viral genome particles per kilogram. If you count how much is this in terms of number of particles in a person of 60 kilograms, it is 7 by 10 to the 15 viral vectors. If you want to target the skeletal muscle, for example, there are approximately 250 million muscle fibers, so you end up at using 7 order of magnitude more vector than the target cells. Compare this with the efficiency of a lentiviral vector, which is, or a retroviral vector, which is 1 vector transduces one cell. So this has ended up in a lot of toxicity in clinical trials. 1/3 of 1,400 people who have received an AV vector for spinal muscular atrophy have developed toxicity, and so we really need to reduce the dose. Let's see if we can find a microRNA or an siRNA that permit to render the cell more permissive to AV vectors. So we perform a screening of a microRNA library to see if there are microRNAs that make cells more permissive, and indeed there are. So these are cells transduced with an AV vector expressed in GFP. Here expressed in DS red in this case. This is why it is red in this case. And this is in the presence of a series of microRNAs from our screening that markedly improve cell permissivity. While the whole gene therapy field works at improving the vector, we target instead the cells. So we want to make the cells for 24, 38 hours be more permissive to these AV vectors. And in fact, we are targeting hits that have nothing to do with the virus entry, but what happens after virus entry. In fact, our microRNA work as sort of adjuvants for therapy, like adjuvants in vaccines improve the effectiveness of vaccines. We have an adjuvant that improves AAV gene therapy. They work with those serotypes, so AV2, AV5, AV6, and AV9. microRNA can even change the type of tropism. AV vectors are poorly tropic for endothelial cells. If you treat these cells with some microRNAs, then you make these cells very significantly more permissive. We screened even a whole genome siRNA library to find adjuvants for permissivity, 18,000 genes, and ended up in a few siRNAs, in this case, that you can inject together with an AV vector in the liver. This is a normal permissivity of the liver to AV8 vectors, and this is the same vector, same dose of vector in the presence of siRNAs against these factors here that emerge from our screening. There are factors that target the processing of AV vectors in the nucleus. AV vectors are single-stranded and needs to be converted to double-stranded, and so you can work on the cellular proteins that will act in the nucleus or that work in endocytosis. This is a normal transduction of cells with an AV-GFP, and this is the same dose after you direct endocytosis toward certain pathways. Another problem in cardiology is gene editing. Very difficult to do gene editing in the heart because you need, in most instances, precise gene editing. Some of the inherited cardiac conditions, cardiomyopathies, basically you cannot do gene replacements, so overexpress cDNA because the disease is autosomal dominant or because the cDNA is too big to fit into an AV vectors. And you really would need to do precise gene editing. But precise gene editing is complicated because homologous recombination is very inefficient in cardiomyocytes. Prime editing doesn't work very efficiently. Base editing is limited to specific nucleotide corrections. So we worked to see whether there are microRNAs that improve precise gene editing, so precise corrections of mutations. So we developed a method for screening for homologous recombinations and ended up in a series of microRNAs that out of 2,000 that we tested, there are 21 microRNAs that increase homology-directed repair significantly, and 10 of these microRNAs have exactly the same seed sequence, which is reported here, and having exactly the same seed sequence means that we screen for a conserved function. So basically, we know that if you take these microRNAs and we give it to cells, we improve the recombinogenic capacity of these cells. So a cardiomyocyte doesn't do homologous recombination. If you give the microRNA for 48 hours, it activates a series of proteins for homologous recombination. So for example, MR11 or RAD50, MBS1, which is part of the mRNA complex that drives DNA repair through homologous recombination, RAD51, which is the main single-stranded binding protein in cells. These are never expressed in cardiomyocytes. They start to be expressed after you give the microRNA. And you can take a CRISPR-Cas9 suite, deliver it to the heart. It does very little homologous recombination. This is homology-directed insertion of a reporter downstream of a cardiac-specific gene. So where you see green, this is homologous recombination. But if you do the same in the presence of this microRNA, you see that a large number of cardiomyocytes in the left ventricle repair a double-strand break induced by CRISPR-Cas9 into homology-directed repair. You can do, you can find microRNAs that increase prime editing. Prime editing is a very interesting technology in which you have a Cas9 molecule fused to reverse transcriptase. It was developed in the David Liu laboratory a few years ago. It works quite poorly in cardiomyocytes, but if together with the Cas9-RT and CRISPR, you also give one of these two microRNAs, you see that it improves a lot. So basically, we like this idea, and this is my last slide, that we can use small RNAs as adjuvants. As I said before, as vaccines needs adjuvants to work, there are several instances which is especially permissivity to AAV vectors and gene editing, which you want a transit effect and a boost in efficiency, and we believe that small RNA can provide this boost. This small RNA can be identified through screening, which is an asset for this technology, and obviously the same concepts also apply to small RNAs that can drive proliferation. We have now tools to deliver them directly to the heart through catheterism, and we have molecules like lipid nanoparticles for non-viral gene transfer, which make the whole combination a very attractive idea to develop RNA, finally RNA therapeutics for the heart. A lot of people involved in this work that I briefly summarize here. I won't name any one of them, but this is my group at the King's College London, still collaborate for AV vector production with the AV vector unit facility that I set up in Trieste. All the animal work in pigs is performed in Italy, in Pisa with Fabio Recchia. And I thank you very, very much for your attention.
Thank you very much.
Congratulations for your optimized microRNA and lipid nanoparticle delivery.
Thank you.
Turn back the clock to remuscularize into the ventricle. I remember you did in the pig using the nanoparticle and microRNA 199. Was successful to regenerate the muscle of the pig?
No, we are testing the pig with the lipid nanoparticle now, what we did in the past with the AAV vector. With an AAV vector, it is super successful, but then the animal, after 2 months, they hyperproliferate and develop arrhythmias.
So the very impressive remuscularized the pig heart
It's an AAV
with
vector.
myocardial infarction, but the pig all died of arrhythmia.
But
so
no, not all died, but several of them developed arrhythmias, some died, yes. Yeah.
So what
but
is
that is an AV vector. AV vector expressed forever.
Yes.
I don't think one can conceive a pro-regenerative therapy with an AV vector.
What is your strategy to overcome the lethal arrhythmia that was
there are no arrhythmias until 7 weeks from treatment.
I see.
And so a lipid nanoparticle with a microRNA that lasts for 10 days, 12 days, we hope it will be sufficient to drive proliferation in the absence of arrhythmias. This works beautifully in mice. Now we are testing it in pigs.
Thank you.
Thank you.
Super interesting, Mauro. I was wondering, sorry, this activation of homologous recombination with microRNAs, how does it mechanistically work? I mean, how do you achieve expression now of enzymes required for homologous recombination? So what do you suppress that enables expression?
We have a mechanism, and I can tell you later. So basically, in brief, what happens is that the microRNA downregulates a repressor of expression of a series of genes that are required for homologous recombination. One of these is a cohesin gene, so one of the genes that is required to take a template for homologous recombination in close contact with the target DNA. This cohesin is never expressed in cardiomyocytes, but becomes very high after you give the microRNA.
Great, thanks.
All right. Thank you so much for the great talk. I was wondering to go back to a little bit of what Jay was touching on with the delivery vehicle in your lipid nanoparticles. You know, they come in a wide variety of molecular weights, and I was wondering, you know, in some other applications with skeletal muscle, the really high molecular weight lipid carriers have been successful. I was wondering if you could provide a few more details about the nanoparticles that you're using here to achieve more optimal delivery.
Yeah, the optimal nanoparticle we have so far is a nanoparticle that involves more cholesterol, the SPC, and a PEG lipid. The most important part is the ionizable lipid, and this is a lipidoid with a quaternary ammonium that is very efficient at binding nucleic acid at pH 4, then becomes neutral, and can create particles that are quite small, so 60, 70 nanometers, which also permits probably diffusion in the heart the way I saw.
So that would imply that your pegylation is actually pretty small on that lipid. Is that right?
Pegylation is approximately 5%. It's not very different from patisiran or the two vaccines. So the key here is the ionizable lipid.
Right.
I forgot to say that obviously if we compare for remuscularization, so for regeneration cells, patches of a lipid nanoparticle, the obvious advantage is in terms of health economics. So we listened to fantastic clinical trials in tens of patients, but we have to develop a method that works in 75 million people in the world with heart failure. And the vaccines has told us that a lipid nanoparticle can be stored, distributed, and administered to hundreds of millions or more people. So it is, should it work, it would be a fantastic drug that can any interventional cardiologist can administer.
So last question, quick answer.
Yeah, thank you for impressive talk. I want to know whether the I'm interested in you make the cells permissive to every transduction using the siRNA and mRNA. So I'm wondering whether they are cell type specific, or we can apply any cell types in the body.
No, what we learn in this journey is that there is no way of having a lipid nanoparticle that are cell type specific so far, except for the liver. For the liver, there is a way of targeting by adding a targeting a molecule, a sugar, which is GalNAc. There is nothing for the heart. I don't believe there will be shortly a targeting molecule for the heart, but the heart can be reached by catheterism. The other lesson that we learned is that different lipid compositions are required to better have transfection of different organs. So those, for example, if you use a patisiran, which is based on MC3, in the heart you have a 3% transfection, but it works well in the liver. While this one we developed is 90% in the heart. So you have to tailor that to your specific organ.
Thank you.
Thank you.
Okay, thank you very much.
Thank you.
So let's move on to the last speaker. The last speaker is Joseph Wu, director of the Stanford Cardiovascular Institute, and Simon Starzer, MD professor and medicine and radiology, Stanford University School of Medicine. The title of his talk is Stem Cells and Genomics for Clinical Trial in a Dish. So, Joseph Wu, please.
Okay, so, thank you, Kaiichi, for the introduction. I know I'm the last person before you guys have a break, so I'm going to quickly fly through some of these slides. And so, again, congratulations to Kaiichi and Jay for organizing this wonderful meeting. So for the next 15 minutes or so, I'm just going to give an audience here an overview of how we use iPS cells and genomics for modeling cardiovascular diseases in the context of drug discovery and clinical trial in a dish. And so I just want to start out by highlighting the Stanford Cardiovascular Institute's iPS cell biobank. I think with the support of Danny Buxton and the NHRBI, over the past 70 years, our lab has been able to make iPS cells from more than 1,800 patients with different medical history, ethnicity, and sex. And then we use PharmGK to help us understand how human genetic variation impacts drug response. We're also working with FDA on drug safety testing, and also have a very robust sharing resource plan so that if any one of you is interested in these iPS cell lines, please reach out to us. Now, from these diverse patient populations, we can now elucidate the mechanisms of various cardiovascular diseases. So, for example, over the past 10 years, we published an iPS cell modeling of hypertrophic cardiomyopathy, dilated cardiomyopathy, left ventricular non-compaction, ARVD, and lung QT. We've also published on modeling secondary cardiomyopathies such as chemotherapy-induced cardiotoxicity. You can also use these iPS cells to study polygenic diseases such as coronary artery disease, hypertension, congenital heart disease. And lastly, you can also use these iPS cells to elucidate the mechanism of single nucleotide polymorphism, or SNP, which I'll, in the context here, I'll actually talk about this. So in terms of the single nucleotide polymorphism, we've been interested in the SNP, or aldehyde dehydrogenase. This is one of the most common genetic polymorphism in the world. It's also quite fitting for today's presentation because we are in the city of Kyoto, Japan, where I bet you about 35% of us among East Asians are missing this enzyme, and 8% of the world population. It causes an alcohol flush reaction, which include flushing, as shown here, nausea, headache, and tachycardia. Antabuse, or disulfiram, works on similar mechanism and is used to treat alcoholics. Basically, they have the same symptoms as this right here. For example, I'm actually a heterozygous carrier for this enzyme deficiency. So my father, he can drink a whole bottle of wine without any problem. My mother cannot drink. So if I actually drink about half a glass of wine, I will have this facial flushing just like this gentleman here. Tachycardia, heart rate goes from baseline 50s to about 120s. So for tonight, I'm actually curious to see how many of you will manifest this right here after we drink. Now, interestingly, this aldehyde dehydrogenase polymorphism involving glutamic acid to lysine switch, this has been linked to increased coronary artery disease, more severe outcomes after heart attack, and also a higher incidence of hypertension and cancer. And so this actually piqued my interest. And so, for example, this slide shows the enzyme deficiency is well documented among Japanese men, Korean men, and also Han Chinese in terms of a risk factor for coronary artery disease and worse outcome after myocardial infarction. And so about 8 years ago, we modeled the worst outcome after myocardial infarction And to model this, we recruited 10 Stanford undergrads who are of East Asian descent. 5 of them are wild type, and 5 of them are heterozygous, which means they'll have similar symptoms like I do. Now, if you're a homozygous mutant, which is about 1% of the population, the symptoms get even worse. And so here we modeled the myocardial infarction by exposing the cardiomyocytes to hypoxia. As you can see here, the wild type cardiomyocytes tolerate hypoxia quite well, whereas the heterozygous cardiomyocytes have much more apoptosis. So overall, this is a schematic. What we think is happening is that after a heart attack, there'll be tons of ROS that gets activated. And if you're deficient in this enzyme here, you have a harder time getting rid of the ROS, and hence you might end up with more cell death and perhaps explaining for the worst clinical outcome that are observed in epidemiology studies. Now, the last study I showed you focuses on the effects of hypoxia on iPS-derived cardiomyocytes to model myocardial infarction. And so as a follow-up study, we recently used iPS-derived endothelial cells from these patients and exposed them to alcohol to model vascular dysfunction and also the risk for coronary artery disease. We know from larger GWAS studies using the Biobank Japan data set that the ALDH deficiency is one of the strongest SNPs that are associated with CAD. And I think mechanistically, this is because alcohol is broken down into acetaldehyde. Acetaldehyde is quite toxic. The enzyme aldehyde dehydrogenase takes acetaldehyde and breaks it down to acetic acid, which is non-toxic. So if you're missing this enzyme, you have a buildup of acetaldehyde, which causes the blood vessels to vasodilate. That gives you the Asian glow. And also the acetaldehyde also causes vascular damage and DNA damage. Now, interestingly, when we did a compound library screen and also molecular docking analysis, we showed that this drug, SGLT2 inhibitor, empagliflozin, which I think the cardiologists here are quite familiar with, it can actually mitigate the vascular dysfunction. And mechanistically, EMPA activates the ALDH-induced oxidative stress and also the nitric oxide reduction by inhibiting the sodium hydrogen antiporter and also activating the AKT-ENOS pathway, a pathway that Dr. Zhao has worked on before. These findings were also confirmed on mouse models of diabetes and also alcohol exposure, showing that EMPA can ameliorate this endothelial dysfunction. Now, so these are 2 papers using race-specific iPS cells to study the mechanisms of alcohol flush variant among East Asians. I think in the next few years, undoubtedly, you'll see more and more examples of investigators using iPS cells in different ethnicities to study biological differences. For example, from a health and science perspective, my lab is interested in understanding why African Americans have much more incidence of hypertension, why Hispanics have much more diabetic vasculopathy, and also why South Asians, for example, Indians and Pakistanians have much more metabolic syndrome. So I want to switch gear to the second part of my talk, which is using these human iPS cells for drug screening. And I think, as you know, this process is actually quite time-consuming and very expensive. For example, it takes about $1 to $2 billion and about 10 to 15 years to bring a drug to discovery, to the clinical trials, to the market approval. So this is actually a major focus of my lab. And so one of the goals in my lab is to start creating what we call a human encyclopedia, a human encyclopedia of signatures for drug responses. And so shown here are 2 examples whereby we assess the effects of 4 different kinds of calcium channel blockers, nifedipine, amlodipine, diltiazem, and verapamil, using iPS cell-derived engineered heart tissues, taking the page from Wolfram and also Thomas' expertise. We measure the functional readouts such as contractility and also calcium kinetics of these EHTs and found drug-specific as well as line-specific changes. And then similarly, we also study 4 different kinds of statins on the right here. These are atorvastatin, lovastatin, simvastatin, and fluvastatin. And so what is interesting to us is that you will see different gene expression patterns among these different drugs. And this suggests that even though they belong to the same class of drugs, they may actually exert very different effects on different individuals. So we'll continue to work on these projects as we try to develop a comprehensive roadmap of pharmacogenomic responses in large cohorts of patients using iPS cells as surrogates. So in a more recent study, on a follow-up study on the statin story, here we use iPS cells and multi-omics to dig deeper into the roles of a statin in vascular health. I think as some of you know, statin is given out to about 40 million adults in the US. I myself, I actually take statin. I take atorvastatin for a high LDL. But there's still a lot we don't know in terms of what is the pleiotropic beneficial effects of statin beyond just lowering cholesterol. And so here, using a TAC-seq, we observed changes in gene accessibility in these iPS cell-derived endothelial cells that were treated with simvastatin. And more specifically, we found regions that were linked to the YAP-TET motifs. These motifs were actually less accessible when you treat it with the statin. And then when we look at the downstream pathway of statin target, it turns out that the HMG-CoA reductase, we found that the YAP downregulation was not related to this cholesterol inhibition, but actually related to an enzyme called geranylneotransferase. And interestingly, when we mimic the type 2 diabetes conditions in iPS cell ECs, we found that the statin can actually protect these cell or the endothelial dysfunction by epigenetic regulation of the YAP, which YAP, I think, as you all know, controls the endothelial to mesenchymal transition. And then finally, we also validated these findings in the mouse models of diabetes and confirmed the in vivo protective effects of statin. And the schematic, again, is summarized here, showing how statin has this pleiotropic effect of protecting endothelial dysfunction through the YAP inhibition pathway. Now, I think in my opinion, the drug discovery application will significantly increase in the next decade. And one major reason is because of the remarkable progress in the AI machine learning technologies. For example, here working with computational biologists at Greenstone Biosciences, we routinely use AI machine learning softwares that allows us to analyze the protein structure by in silico molecular docking. Once we know the molecular target and the protein structure of the drug or the disease that we want to go after, we can actually do a virtual screen of about 1 billion compounds at a time. This will take us about 2 to 3 weeks to screen through them. And once we come up with the drug hits, instead of testing the mouse cells, we'll then test them on the patient-specific iPS cells that have the exact mutation or the exact disease that we designed the drugs for. So I think this is actually going to be much more accurate and also clinically relevant. And in case you're wondering here, this video actually shows that 3 drugs that bind to the protein target that we're interested in. And here you can see that the drug in pink color binds to the target, whereas the other 2 drugs do not. And so the drug in pink color will be something that we would chase after rather than the other 2 drugs. And so just want to give you an example of how we use AI machine learning in silico docking for drug discovery. I think, as some of you may know, in the US, marijuana is legalized and it's being increasingly used. And interestingly, the UK Biobank database shows that marijuana use is associated with myocardial infarction, but the exact mechanism is not well defined. And here, we show that the delta-9 THC, this is a psychedelic component of marijuana. It can actually cause vascular inflammation and oxidative stress. And we confirm this by using iPS cell-derived endothelial cells. We're confident that the mechanism is through binding of the THC to the cannabinoid 1 or CB1 receptor, which activate the MAP kinase, and which then causes oxidative stress, inflammation, and also cell death. So importantly, as I mentioned, using in silico chemical library, as shown in here, we were able to show that genestin, which is an isoflavone, actually serves as a neutral CB1 antagonist, essentially inhibits the, basically competes with the THC from binding to the CB1 receptor. And these findings were also confirmed in these iPS cells. For example, when you have the iPS cell exposed to THC, as shown in blue here, you have increased inflammation, whereas if you treat it with genestin, you actually have decreased inflammation. And finally, the drug targeted for ginestin was also confirmed in 2 independent mouse models of corneal and carotid atherosclerosis. So as you can see here from these examples I just showed you, we're integrating iPS cell biology with cellular, molecular, and functional readouts, and also multiomics analysis to help us better understand the disease at a systems biology level. I think in the next few years, you'll see more and more examples of investigators are using large-scale human iPS cell populations to understand, for example, expression quantitative trait loci, EQTL, and also to study the genotype-phenotype relationships via these co-expression networks. And so as an example, we're now working with the FDA to study 200 patients, cancer patients, which we generate the iPS cells. We pool them together at 20 lines per what we call a cell village. This allows us to expedite the workflow and expose them to increasing doses of the doxorubicin here. As long as we know the DNA sequencing of each patient, we can then use a single-cell RNA-seq to deconvolute which data point comes from which patient. This allows us to dramatically increase the throughput. It also allows us to elucidate the EQTL related to doxorubicin. I think this approach is actually probably more accurate than the traditional clinical GWAS studies that is actually a lot of times quite difficult to assess due to the variability in terms of access to healthcare by our patients, differences in socioeconomic status, and also the simple fact that when you prescribe a drug as a doctor, 35 to 40% of our patients actually do not take the drugs. So it's very difficult to do these type of GWAS studies. Here, everybody's locked up in a cell incubator. We get them the same dose and see what happens here. And so for the last part of my talk, I just want to touch upon how we can then use these iPS cells to implement this concept of a clinical trials in a dish. This is a concept that I've been talking about for the past 10, 15 years. Here, we should be able to recruit patients, generate the differentiated cell type of interest, test which patients respond to drugs and which patients do not by using the iPS cells as a surrogate, and then go back to perform the actual drug study on the patients who are positive responders to the drug. And that way you have a higher chance of success rate for your phase 1-2 clinical trials, decrease the cost, and also accelerate the timeline of drug development. And so as some of you may know, the FDA Modernization Act 2.0, this was recently passed by both Senate and House on December 2022. This law authorizes the FDA to use alternative testing platforms such as stem cells, organoids, and also in silico modeling, and for some drugs to completely eliminate the requirement of animal testing. The goal here is to improve the predictiveness of preclinical and safety and efficacy assessments, and also to reduce the number of animals that are used for drug discovery and development process. And I think some of you may also know that the EPA, the Environmental Protection Agency, plan to completely eliminate all animal testing by 2035. So we believe the FDA Modernization Act 2.0 will pave the way for the next era of drug discovery that uses AI machine learning, essentially predictive AI, also generative AI, to find the optimal candidates, for example, drugs, peptides, antisense oligonucleotides, RNA therapeutics, and so forth. You can then test them in vitro, followed by your in vivo PK/PD, ADMET studies. Afterwards, a clinical trial in a dish model can then be established to predict ideal clinical trial design prior to giving the therapeutics to the patient. And so now at Stanford, I just want to tell you a quick example of a clinical trial in a dish. About 6 years ago, we were referred this large family in which this family has lamin AC, a frame shift mutation, which leads to early termination. Lamin cardiomyopathy accounts for about 10% of familial dilated cardiomyopathy, so roughly about 1 in 20,000 patients. In this large family, many of them had cardiomyopathy, atrial fibrillation, atrial ventricular block, and 8 of them actually died from sudden cardiac death already. And so using these iPSL cardiomyocytes, we confirmed that the lamin mutation is indeed responsible for the cellular arrhythmia and also the impaired contractility. And so this cartoon here highlights what's going on. Typically for the healthy cardiomyocytes, you have normal expression of the lamin protein, normal nucleus structure, and also normal lamin-associated domain. But then the patients with the lamin AC frame shift mutation have a normal structure that causes increased open chromatin in the lamin-associated domains instead. This then leads to activation of genes that should have been silenced in otherwise healthy conditions. And using RNA-seq and ATAC-seq, we were able to show that one of those abnormally activated genes actually involves the PDGF pathway. And then we also, for this study here, we also screened for FDA-approved drugs and found that a tyrosine kinase inhibitor at a low dose, quinololip, can actually be used to reverse some of the abnormal phenotype. Now, what's interesting is that we noticed many of these same laminin patients within the same family develop early onset hypertension at about age 30s and 40s. And so here we decided to take the iPS cells, differentiated the endothelial cells instead, compared to the healthy versus the disease, and then compare the differences. We perform a whole battery of functional assays and identify KRF2 as a potential transcription factor that is responsible for the endothelial dysfunction as outlined in this cartoon. And again, going back to the FDA-approved drug library screen, we found that lovastatin is actually a potent KRF2 agonist, which can be used to rescue the endothelial dysfunction in vitro. So we now want to test to see if this is true or not. And so we actually, the same laminin patients whom we tested the iPSL ECs were then brought back to our clinic, and they were given lovastatin daily, even though they don't have cholesterol issues. The clinical data is shown here. We use EndoPAT, which is a machine that can measure flow-mediated dilatation. Normal index is 1.67 and above, and as you can see here, laminin patient number 2 started with a poor index of 0.48, improved significantly to 1.06 after 6 months of a lovastatin treatment. So I think this is also a clear example of what we call clinical trial in a dish, whereby we first identify a large family laminin mutation. We then screen for a panel of FDA-approved drugs using their patient-specific iPS cells, and found that lovastatin can be used to improve the cellular phenotypes by enhancing the KRF2 pathway, even though they don't have high cholesterol. We then confirm these in vitro findings in the actual patients themselves by the EndoPAT measurements. So in summary, I believe the paradigm shift in the future of precision medicine and also drug discovery and also clinical trial in a dish will involve these patient iPS cells to help us better understand the molecular mechanism at first. These iPS cells can then be differentiated to all these different cell types here. We can then link them to the clinical genomics as well as the multi-omics sequencing. We can also use AI machine learning for molecular docking, molecular dynamic simulations, in silico ADME predictions. And by combining these three here, hopefully we can come up with the drug hits that are much more precise, and then go back to test it on the patients that have the disease that you designed the drug for, and therefore completing this loop of clinical trial in a dish. And so this concludes my talk. I want to acknowledge my postdocs, my instructors. Some of them are looking for jobs, and the biobank staff, my former postdocs whose work I highlighted here, my collaborators and funding support here. Thank you very much for your attention. Yeah.
So the paper is now open for discussion.
Yes, that's it.
Joe, absolutely fantastic talk, as always. Can I ask you for your experience of completing that circle? You show very nicely about how to get mutation-specific or even patient-specific drugs that are effective in the iPS model. But could you elaborate a bit more on how you take that back to patients? What's been your experience in terms of treating patients, and what are the sort of roadblocks to that? I think that's a particular challenge.
Yeah. I mean, that's a very good question. I think right now the roadblock is really the cost and time. So we can't really do this for individual person just walking in the street, just not possible. I think for those studies, these are patients that we refer to our clinic, and large family, we look at it and say there's a mutation, there's low-hanging fruit, we can go after it, we can find what's going on. Keep in mind, again, this is just my own personal opinion, keep in mind that I think probably genetics account for about 10% of how we manifest disease. Probably the other 90% is how we carry ourselves every day, and sometimes it's really hard to capture that using these iPS cells. But for now, I think the clinical trials in a dish is one example, particular disease. What we're actually more interested in is a large scale. For example, I develop a drug, I give my drug to iPS cell-derived brain cells from everybody in this room, and all the patients have some type of disease, and we're trying to understand what is the population scale of how people respond, because traditionally, drug companies have been fixated on bar graphs of A versus B. A means drug, B means no drug. It's very skewed because you're using the same cell line or the same patient. And here, in that case, you're just measuring the technical variability of the technician pipetting. Here, if I do it on everybody, I'm actually understanding the genetic variability, and that's why we're very much interested in our lab. Yeah. Yes, Adam. Yeah.
So, mic one, please.
Sorry. I was wondering, specifically with your AC patients at the end, when you see that frame shift, is that causing a truncation such that the lamin itself doesn't form a network, and that's the reason why the LADs don't bind correctly?
Yeah, so basically it's a frame shift mutation, and that protein is completely gone, and then the patient also has one extra, one quote-unquote healthy copy, and I think it's more of a haploinsufficiency issue.
Okay.
Yeah.
But then, so do you see global rearrangements in like ATAC signatures?
Yeah, so I think not just us, but I think, for example, Raj from UPenn, he's taken some of our cells, and he's actually gone much more deeper in terms of the epigenetics and show changes on the epigenetic level as well. Yeah.
And so specifically then, do you see any differences in things like cardiac transcription factors or?
Oh yeah, so the paper actually has all the RNA sequences. You'll see differences on the RNA transcriptomics. You'll see differences on metabolomics as well, just with a simple frame shift mutation of the lamin.
Mm-hmm.
Yeah.
Thanks.
Yuji?
Yes.
Very good presentation as always. So I have a question about aldehyde dehydrogenase. Actually, I obviously don't have this enzyme. And I was wondering, for instance, if I keep drinking, my lead flash reaction will decrease. And so my question is, always some mutation have a compensation mechanism. For instance, aldehyde dehydrogenase case is probably xenobiotic, the reaction will be increased, for P450 or so. So do you think this compensation mechanism affect your screening for?
Maybe I could talk to you offline. If I understand correctly, you're asking why, through the natural evolution, why East Asians have this particular enzyme deficiency. Actually, it turns out nobody really knows why East Asians, but you could imagine that if you're missing this enzyme deficiency, even though this enzyme is involved in getting rid of some of the ROS and also this enzyme deficiency actually causes you vascular inflammation. The flip side is, say, if you're missing this enzyme, it's really hard for us to become alcoholic. Right? I just can't drink, and But in terms of why there's this founder effect, there's a lot of theories, and during the cocktail I could tell you about 3 major theories why why East Asians are missing this enzyme. Yeah. Thank you very much. Yeah. Mm-hmm.
Okay. Other questions? So thank you, Professor Wu, for a nice presentation. I'd like to close this session.